Hi, everybody. My name is Sequoia Burleson Ross, and today I will be talking about the cell cycle and the regulation of such. So, to start off, a friendly reminder of the cell cycle. The cell cycle is what eukaryotic cells go through for mitosis, and these are the different phases. And there are three checkpoints throughout the entire cell cycle. So, as we can see here, if we'd start roughly around here, the cell would start off in the G1 phase and possibly move into the S phase, the G2 phase, and the M phase before finally separating. So, first, the first checkpoint is the G1 checkpoint, and this is the main dividing determination point. Usually, in order for a cell to pass the checkpoint, you have to ensure the cell has to ensure that it is large enough to divide, that it contains enough nutrients to support two cells, and that it has received the positive cues from its environment and from within itself. So environment such as growth factors and within itself, making sure that the DNA is in fact intact and free of damage. Depending on the cell type, there may be additional cues needed, such as mechanical cues from the extracellular matrix. So if the cell does not pass the necessary steps, then it will continue off onto this small loop that we see right here in the G0 phase, also known as the resting phase. But if the cell were to continue on into past the checkpoint and go on to the S phase and into the G2 phase and reach the second checkpoint, the G2 checkpoint, then this is when the cell has to ensure that its DNA has been fully replicated and it is complete. Because if the DNA is compromised at all, then the cell cycle will stop as the cell attempts to either complete the DNA replication from the S phase or repair any of the DNA errors that may have occurred. Now, if the cell is unable to repair the errors, then it may go through apoptosis, which is also known as self-destruction. This mechanism is to ensure that any damaged DNA does not get overlooked and passed on to the daughter cells. This is because it is a cancer preventative measure. Then the third checkpoint within the cycle is the spindle checkpoint. It is within the M phase. During this checkpoint, this is where in the cycle the sister chromatids attach to the spindles, the microtubules, and the separation of this. Once the sister chromatids actually separate within the cell, mitosis is going to complete and occur because it is irreversible once those sister chromatids are separated. So it is essential that all of them are connected to a spindle. And during this checkpoint, it is crucial to make sure that those chromosomes have been attached, because if they are not regulated during the like within the actual metaphase plate, rather it is found for the straggler chromosomes to be found in the matrix. Therefore, the cell cycle has to stop while spindles find and reattach to those chromosomes that are left hanging pretty much. And they're pulled to the polar ends while the cell completes its mitosis. All right, so now there are other regulatory proteins and enzymes needed for the cell cycle to continue. Geez, sorry. Okay, so as we can see here, I have a graph. Down here at the bottom, we have the different phases within the cycle of the cellular cycle. And this shows concentration, right? So there is a protein called cyclin, and it is pretty much one of the really more important regulatory factors within eukaryotic cells. As we can see here, there are 4 different types, at least within mammals and humans, and they seem to be correlated to the phase that they occur in, except for G1 here. As we notice, it is spread across the entire cell cycle, just going down for mitosis and reinitiating within the daughter cells. That is because the cyclins are in a low concentration when they're not needed, but as they become needed for the role that they play within the cell, their level of concentration increases. So, for example, the M cyclins are responsible for the nuclear envelope breakdown and the chromosomal condensation that's interesting. Where they condense. The amount of cyclin proteins present within the cell varies based on, again, their role and their job and how when they are needed. As they're needed, they increase, and otherwise their levels are very low. So for the M cyclin, where it peaks at the transitional point, right after from the G2 phase into the M phase, this is because it is needed more so in the M phase for, again, breaking down the nuclear envelope and interesting. Now that I've said that out loud. The G1 cyclin, however, is again present throughout the entire phase because it is utilized throughout the entire cell cycle. Okay. Now, there's an enzyme noted as the CDK. Stands for the cyclin-dependent kinases. Hope I said that right. So CDKs are inactive until a cyclin attaches to it, so it activates it for it to be able to perform its job by targeting specific proteins and marking them. Basically, this mark is the phosphate group that attaches to the proteins. This process is referred to as phosphorylation, or where the target cells are phosphorylated. Here's your phosphate group. So that's what's attached to all these target cells, and that then activates the target, not the target cells, excuse me, the target proteins. And this phosphate group activates the target proteins to then go ahead and do their job. So, for example, in this diagram, we have the G1S cyclin. And this will now initiate the S phase to begin, where the DNA replication enzymes are activated because this enzyme was able to activate them and, the domino chain reaction. Without the activation of the cyclin, though, the CDK is inactive and therefore is not able to do its job by marking these proteins and activating them. Therefore, this is a part of the regulation that would prevent the cycle from continuing. Let's see. For another example, with the M cyclin, okay. So, since the role of the CDK is an enzyme that has to be activated by one of the cyclin proteins, the CDK enzyme itself remains pretty much consistently at a constant level because it can be reused by any of the cyclin proteins that activate it. Those specific proteins are what activate the next group of proteins to be activated, essentially. So this chain reaction, if we were to look at it from another cyclin perspective, then having the M cyclin actually creates a compound that we can call the maturation promoting factor. This is also initialed as the MPF. So the MPF complex was initially noticed and observed in the 70s and officially discovered in the 80s when it was first noticed within frog egg cells. As the cell was stuck in the G2 phase, it was able to be forced into the M phase due to the presence of these noted MPFs. That is, again, because like we learned with the last cyclin, it is activated and the level of the cyclin needed only increases when its role is needed and the cell is able to continue on with its cellular cycle process. So it now phosphorylates new target cells, different ones, allowing for the M phase to begin, where the spindles form, the chromosomes condense, and the nuclear membrane breaks down, like I said before. Over here, we have an example that demonstrates how it marks and disassembles the nuclear envelope. So, normal cell with nuclear envelope, the CDK and the M cyclin compounds, also known and referred to as the MPF. This then adds the phosphate group to the proteins needed to help disassemble the entire nuclear envelope and condense the chromosomes. Without this process, the cell would not be able to continue on with onto the next phase. Okay, let's see. And so then, okay. I don't know why it does that. I click it and it won't click. Okay. Now, another important feature about mentioning the MPF, that CDK with the M cyclin, is that it also will activate the anaphase promoting complex cyclosome, also known as APCC, as we see here. This actually destroys the M cyclin and anaphase. It will also allow for the cell to be able to complete its mitosis process and for the new daughter cells to be able to properly enter into the G1 phase on their own. The APCC also destroys the proteins that bind the sister chromatids together. Now, it doesn't directly go in and destroy them. As we can see here, there's a process. The APCC is also an enzyme similar to the CDKs, but instead of phosphorylizing a protein, it actually it tags the proteins with ubiquitin, also known as UB. So as we see here, the APCC marks the securin with a ubiquitin. The securin is the protein attached to the enzyme separase. When they're bonded, separase is inactive. As soon as that UB attaches to the securin releases from the enzyme and is actually sent into the protosome for being broken down and destroyed. The separase then moves over here because now it's active. It actually targets directly the proteins holding the sister chromatids together, known as cohesin. It breaks this apart and allows the sister chromatids to separate. Excuse me. Okay. So while cyclins and CDKs and APCCs are direct regulators of the cellular cycle, they have to be signaled by cues. And these cues can either be internal cues, such as damaged DNA, or it can be an external cue, such as growth hormones. And these would increase the CDK activity. When DNA is damaged, it is the cell's responsibility to find and fix the possible damage. Otherwise, it cannot continue on with cell division. One of the main proteins that help with this is the P53. It is also well known as a tumor suppressor, and it will stop the cell cycle if the DNA is damaged. At the first checkpoint, if the DNA is damaged, then the P53 will trigger the production of a CDK inhibitor. These proteins are referred to as CKI. So like I mentioned before, I believe I did this time, at least there is the possibility of the CDK, even though it is activated by the cyclin, if it has another bond, then it is inactive. Oh my. Sorry, didn't realize with the lag. So here's my p53, the damaged enzyme, and it produces the CDK inhibitor proteins, which then attach to the CDK with whatever cyclin it is. So for example, we were discussing the G1 phase where we first check to make sure that the DNA is not damaged. So this would then pause the G1 cycle while the cell tries to attempt to correct the errors. Now, if it cannot correct the errors, and and if p53 is for some reason either inactive or not present, then okay, so then that would create a problem because the cell cycle would not stop due to damaged DNA. It would continue through its cell cycle. And we don't want this because, like we mentioned before, we want to regulate it to make sure that we're preventing cancer and preventing those mutations from spreading onto the daughter, well, mutations from, yeah, being passed down through the lineages. So, cancer is a disease that is characterized by the uncontrollable division of cells. Normal healthy cells, like we see over here, if we were to put normal cells and cancer cells within petri dishes, as we see here, normal cells would require growth factors in order to continue the cellular cycle and actually divide through mitosis. Without growth factors, there's no division, there's no growth. They just stay their normal selves. Cancer cells, on the other hand, have a lot of mutations that actually allow for no regulation, and they can grow and divide with or without growth factors. In fact, it's just, I don't know, it's insane, all the different ways that cancer has been definitely mutated to just constantly be multiplying. Normal cells would stop dividing from contact with each other because they ran out of space, but cancer cells, no, they continue to divide. And this is what creates the dense, clumpy, layered tumor. So cancer cells are kind of also referred to as having replicative immortality because while normal healthy cells can divide about 40 to 60 times before dying, cancer cells can just keep on dividing because they express an enzyme called telomerase, which reverses the degradation of the telomerase ends that would normally wear down from the constant replication of cell division. Cancer cells are unlike normal cells because they can travel through the body, and this process is called metastasis. Cancer cells promote the growth of new blood vessels, also known as angiogenesis, but these blood vessels are meant to bring oxygen and nutrients to the tumors to help it continue to grow and divide. And there's the odd phenomenon that cancer cells fail to undergo apoptosis. So instead, they've changed their metabolism to support additional division. It is theorized that there has to be a multiple step system of mutations in order for this to occur due to the variety of mechanisms that have failed for preventing these cells from being cancerous. The series of mutations that would be required would increase the division speed, avoid internal and external regulators and signals, never mind the ability to evade apoptosis. So that many mutations, there's no way it could just be one. So therefore, it is first assumed that there has to be a loss of the cell cycle inhibitors to allow for this rapid mutation and the passing on of mutations. Then the overgrowth of the cluster at first may be considered benign because that not yet has the ability to metastasize. Instead, it stays within its little pocket. It hasn't spread into the tissues yet. Next, a mutation may increase the activity of positive regulators, which would further increase the division and growth of the cells. The tumor is considered malignant when the cells from the group can now invade the other tissues. More mutations may also occur within the genome for the stabilizing proteins that would normally find and actually repair the DNA or maintain the telomere caps, prevent DNA binding chemicals, and like a domino effect. Once one of these major proteins is non-functional, then the rest will mutate and it will just continue to pass on to the daughter cells, leading to rapid mutations and cancer development. Ultimately, there are two ways that cancer development can really develop, either from overproduction of positive regulatory enzymes and proteins, or from the inactivation of negative proteins, the ones that would stop the cycle to help regulate the system. All right. So when a positive cell cycle regulators are overactive, then the cells may have growth factor receptors that are triggered without the growth signals, like we had talked about in the Petri dishes, or there may be an abnormal high amount of cyclins. Those would activate all the CDKs to continue the cycle. The genes for these processes are called proto-oncogenes, but once they are mutated and overactive, then they're just called oncogenes. Such mutations can be a change in the protein's amino acid sequence, which would alter its tertiary structure, leading to a possible growth factor receptor protein, such as the RAS protein, to be left in its on state. So like when it normally is triggered by growth proteins, is on. And if it doesn't have it normally in a normal cell, it would be off. But with cancer cells, it doesn't matter. It's always on for some reason, allowing the cell division to keep occurring. The RAS is actually a G protein, and it affects the RAF enzyme. Excuse me. This would mean that one mutation would affect them all. Let's see, mutations involving regulatory proteins, such as tumor suppressors, such as the P53. Not having these, or not, either them being non-functional or them not being present. Like I said before, there are three pathways that they go through and help prevent cancer by preventing the cellular cycle to continue. Like I said before, they would halt the checkpoint or stop the cycle. They would then find and repair the DNA. But if they can't repair the DNA, then apoptosis is triggered. If that's not present, then there is no apoptosis, and then the mutations get passed down to the daughter cells. Oh, alrighty. Anyway, that is my presentation. If you have any questions, please feel free to reach out and leave any comments. Thank you so much for listening, and I hope you have a wonderful night.